targeted recruitment and retention, including faculty from ecology, evolutionary biology, geoscience, water science, and chemistry. And doing research on management of the natural and range resources is just part of this mission. So I'm extremely grateful to be part of the desert ecology research team. And today you're going to meet three of us, and we're really excited to present to you our toolboxes that we use in research in this area. So we'll have Matt Petrie, Dr. Matt Petrie presented today. He's our ecosystem scientist. We have Dr. Scott Abella here. He's our restoration and plant ecologist. And I am a soil ecologist that recently joined UNLV a year ago. And we have several different skill sets that are interdisciplinary, and we are excited to work together. So we want to show you a little bit of the research that we're doing and how we can potentially collaborate with you all. All right, and so how's this gonna go? I'm gonna start presenting on the little guys, and we're gonna scale up with having Dr. Abella follow my presentation, and upscaling to the broad scale with Dr. Petrie finishing our presentations. All right, so my talk is focused on soil microbes. What soil microbes can tell us about dryland biodiversity, health, and ecosystem functions. And so let me tell you a little bit first of what type of research our lab does. So we like to characterize and quantify soil microbial diversity in drylands, especially with a focus on cyanobacteria. We investigate the roles of cyanobacteria and other microbes in dryland ecosystem function and soil health. And we like to identify and extract key cyanobacteria from dryland soil microbiomes to be able to enhance soil health. And lastly, we love to raise awareness about the importance of the dryland microbes as an important ecosystem component in our landscape. And where do we do this work? Well, we have mainly focused our efforts in the drylands of the Americas. And so we are particularly looked at soil microbial communities in the hot desert biomes because they're poorly characterized. And I will show you today some case studies from these areas, including the Mojave, Sonoran, and Chihuahuan deserts. Why do we love researching these areas? Well, as you all might know, they contain the most beautiful dryland landscapes. And actually, they also contain a phenomenally diverse nature of soils. And guess what? These diverse soils represent really true treasure chests when it comes to microbial diversity, but it's a treasure chest that remains to be uncovered. And so, as you may know, in general, soils are exceptionally diverse habitats. And we learned that in soils, we can find the majority of all terrestrial life forms. But regardless of the estimates on the vast numbers of organisms that soils can house, there's only a very limited knowledge of the biological species that live there and what each of them actually do. And this knowledge gap is particularly large in drylands. And what we have been experiencing in our lab is that you can actually, once you look at these dryland soils, discover a fascinating world of beautiful organisms that are extremely important for dryland ecosystem health, as you can see from these images of examples. And now, in our rapidly changing world, there's a huge need and urgency to do this kind of research, especially right now. Turns out drylands are actually one of the most neglected biomes for biodiversity research in general, although we know that drylands cover an enormous extent of the terrestrial ecosystems, as seen here in the orange-red shading on the map. And at the same time, our drylands are one of the most threatened biomes, resulting from global change to the rapid human population increases. And with more people comes a huge demand for land for housing, infrastructure, energy, and recreation. And we know with that comes severe environmental problems, such as non-native invasive species invasion, dust emissions, soil degradation, habitat loss, and so on. And a lot of this damage happens at the soil level. And every day we're losing valuable dryland topsoil and continue to degrade it. And it's really that topsoil where a lot of our beloved soil microbes that are important to ecosystem health are housed at. So you get why there's an urgent need to study dryland soils and their microbiota. So let me show you some data that supports how cool and unique these dryland microbes are. So in this study here, we investigated the surface soil microbiomes of the Jornada Basin that's in the Chihuahuan Desert in New Mexico. And we used next generation sequencing data to investigate what abiotic and biotic factors shape patterns in microbial diversity and distribution. So I can't go into detail, but what we found is that the overall landscape features, such as vegetation type that you can see here on the left and landforms on the right, really shape the surface soil microbial diversity, as you see it in the significant difference of the richness values. And that also influenced community composition and structure. So landscapes are actually shaping community composition as indicated by the colored ellipses here with an example of cyanobacteria and fungi. And further, a suite of soil properties explained the relationships amongst landforms, the microbial community composition, and its characteristics. We also been isolating microbes to further study them. And this gives me a great chance to advertise our culture collection at UNLV, which contains hundreds of strains isolated from extreme environments with a focus on drylands. And so here's a cool example of a lineage that we worked on that's been found so far only in dryland biocrusted soils. This is Mojave, and originally, the type was described from biological soil crust in Joshua Tree National Park. And for a long time, for like 2 decades, it was the only record and species known. We've been able to detect it with next-gen sequencing approaches in the past 5 years in Spain and in the Atacama Desert. However, we recently successfully isolated 2 new strains and describe 2 new species. And so this genus with the 3 species that we now have is pretty rare, but it's kind of cool because it forms like very unique ways of being adapted to a dryland system, such as their own sunscreen pigments or resting stages that help them survive. All right, so why should we care about soil microbial biodiversity management view? Well, because they can maybe form intricate co-occurrence or interactions with other microbes, such as we've shown with our collaborators from the University of California, where cyanobacteria were co-occurrent with certain types of fungi. They also can form intricate biotic interaction with plants. So our collaboration with Dr. Abella here, we want to explore how maybe inoculations could promote or affect plant growth and health. And microbial traits can lead to difference in functional performance, which we're excited to look into further with the collaboration of the Petrie lab and the Bowser lab here at UNLV. All right, so dryland microbes contribute to biodiversity as additional members of the community, and they need to be considered in biotic integrity assessments. All right, another really important thing is that we in resource management do not recognize microbes with the same importance as plants and animals. But if you look at the dryland landscape, they're crucial parts of it. just take a look at this image right here. As you know, we have this unique makeup of the landscape. Plants are not continuously distributed, and we have these large patches that are open. And it's in these open patches in between the plants that we can find a very thin layer of topsoil that is just choked with microbial life. If you don't believe me, I got you some data here from the Chihuahuan Desert, where we studied sand sheet that was associated with different dune types along a chronosequence. Again, I can't go into detail, but what we did is we explored the microbial biomass of the first 1 centimeter of soil on the left and the subsequent 2 centimeter of the subsurface soil. And to give you just a comparison here, the blue dashed line is the average soil microbial biomass in an agroecosystem, so in a kind of like a boosted system in our drylands, which has a large amount of microbes in it. So what you can see is really that 1 centimeter, that really thin topsoil, contains an enormous amount of microbial biomass. And the microbes that are in this thin surface layer can often form what's called biological soil crust, or short biocrust. And these are super cool dryland soil phenomena that house a highly diverse set of microbes in it. So these microorganisms build soil aggregates that are living and form like this crust at the surface. And it is the cyanobacteria that actually are considered as ecosystem engineer, creating these biocrusts. And we love them in our lab. And what our research shows that in our North American deserts, biocrusts are actually highly variable in their extent of ground cover, as shown here with the average ground cover values in the different region. But if you go into these places, over half of the ground can be covered by biocrust. And a lot of them, when you look in the hot deserts, are actually dominated by cyanobacteria. And there seems to be also an interesting interactive story going on with climate, paramaterials, and the way the landscape is shaped. For example, in southern Mojave biocrust, they're best developed on goosey granitic paramaterial on young to intermediate aged alluvial deposits, while on gypsum soils, there's just absolutely stunning crusts on the substrate. Oops. Skip that one. So in a nutshell, research in hot desert biomes supports cyanobacteria crust, while they can be pretty cryptic to identify. And another cool finding of our work was that the composition and changes in biocrust can be understood with this soil geomorphic template framework. So linking landscape, soils, and microbes together with impacts to ecosystem function. So how does it relate to management? Well, first, we have to recognize that microbial communities and the biocrust they form can be dominant living ground cover. The static and transient site conditions can be predictive for their diversity and abundance. So we can work with Dr. Abella's lab on that. And they provide ecosystem services that are worth managing for, such as soil stability, nutrient cycling, carbon sequestration, where Dr. Abella and Petrie, we can all work together and characterizing that for the Southern Nevada region. All right, lastly, I want to focus a little bit more on our ecosystem services to show what kind of difference they can make. So soil crusts are actually not all the same when it comes to ecosystem service provisioning. They differ by the processes that form them and the community members that dominates the crust aggregates. But we can actually arrange them along a structural complexity gradients that has an impact to ecosystem functions. So we can group the least complex type on the left with increasing complexity as you move to the right with physical crust, incipient crust, fungal crust, two types of cyanocrust, and with the highest structural complexity in our lichen crust and bryophyte crust. With that structural complexity, you can actually add multiple functions together with the most functions provided by lichens and bryophytes. How does that impact ecosystem services? Well, structural complexity translates into changes in function as shown here with our measurements of soil aggregate strength. So you can see that for aggregate stability, we can get a climate stability as you compare these different crust types with each other. So as you increase structural complexity, you increase soil aggregate stability. However, it also matters what type of above ground structures you have with like lichens and mosses having these distinct above ground structures that in return are more vulnerable to certain pressures such as downward pressures or shear forces. So cyano-dominated crusts can be more resistant, which has implications for management. So here we are, management considerations. So when we incorporated the cover of the different crust types and integrated these three soil strength measures, it translated into really interesting effects on ecosystem function. So this graph here on the left is a little bit complicated. What, in a nutshell, what it tells us is that for dune slopes compared to sand sheets, the microbial communities that form these biocrusts that were more structurally complex really contributed to soil aggregate stabilities. Okay, so the dunes are mainly stabilized by microbial forces and not so much by vegetation. And that matters to the White Sands National Park resource management. Additionally, microbes also impact other things such as porosity and bulk density. So with more microbes in the soils, you get a less dense soil aggregation, so like more fluffiness and more macropores, which has impacts to hydrological cycling and so on. All right, so in summary, what did we learn? Well, we are facing microbial blindness. So some microbes are prominent landscape components in drylands with their own management needs. However, management practices currently are still plant and animal centered. Oh, come on. Okay. Microbes operate from the bottom up, so they have impacts to other organismal groups from the bottom up. Inventorying dryland soil microbes is highly relevant and this work matters. I hope I demonstrated that. Microbes need to be therefore included in biotic integrity evaluations, things like ecological site and state and transition models, woo, and ecosystem service assessments. And we need to consider that our dryland microbiomes face the same numbers of global change threats and that we may lose a resource without knowing it. All right, so I hope this shows you that drylands offer an opportunity for discovery even in the microbial world, and we need you to join us in this effort. And with that, I'm going to turn it over to Dr. Abella, talking about restoration ecology.
All right, in the section that I'll be showing today, I'll be going through a recently published habitat restoration practices synthesis for the Mojave and Western Sonoran Desert. And this was supported by the Clark County Desert Conservation Program and the California Fire Science Consortium for fire science outreach. And we run the deserts portion of that through UNLV. And just want to mention too that the synthesis is freely available. It's up on my website, and I can also email it directly if folks would like to email me. So, first question is, when might we need to deploy restoration, right? We really want to be able to prioritize where it's most needed. And just want to give an example here of some of the really long recovery times that we can see in severely disturbed Mojave Desert ecosystems. So this is an image from Red Rock Canyon right here in southern Nevada. And the left is unburned creosote bush, mature shrublands. And then the right photo was burned by the 2005 Loop Fire. And so this is the extent of the recovery about 15 years after the fire. And so we still have very different habitat conditions with good plant cover on the unburned portion on the left for animals such as desert tortoises, and then much more sparse cover on the right-hand side. So where restoration is desired, what we're trying to do is to hasten the rate of ecological recovery and also to direct perhaps the species composition toward desired functions that we wish to achieve. So in our review, we identified over 50 published restoration papers in the Mojave and Western Sonoran Desert, and we categorized these papers into three major categories, including restoration of site environments, including techniques such as salvaging and replacing topsoil, revegetation, including outplanting nursery grown seedlings and seeding and other treatments, and then restorative management actions such as fencing for protection or reducing non-native plants. And so next, I'll go through a series of slides showing examples of what's in the review and examples of these restoration techniques, including some of our own studies here in the Mojave Desert. So first, active revegetation using outplanting. This has had some failures. So there's been tested in 16 different studies, but in general has had at least some reliable plant establishment in most studies. And it's been deployed to strategically revegetate large disturbances to create revegetated islands, such as shown here on the right. And that's a creosote bush there in the foreground that's in a shelter. And what was really neat about this study is that within a few years, many of the outplants were already flowering and producing seed. And so that can be one of the goals, that we can have self-sustaining populations of the outplanted species themselves to revegetate broader areas. And then on the left is a recent outplanting to strategically conceal the entry point of a decommissioned road. And so, this again, this is one of our goals of outplanting, is to just to deploy this strategically to revegetate areas potentially more reliably than seeding. So there were 16 total studies that assessed outplanting effectiveness covering 46 species, and we summarized which species thus far have been the best performers in outplanting. And then we also summarized effectiveness of different treatments that could be applied with outplanting to try to enhance plant survival. And what we did is we expressed effects of the treatments as a ratio of doing nothing, basically planting a plant without treatments. So like for the cage there, the first row there, the +4, that means that in that particular study, 4 times more plants survived if they were given cage protection from herbivory and other disturbances. And there's an example of that on the right-hand side of the slide, enclosing a globe mallow. And in fact, caging and sheltering was one of the most effective treatments for increasing survival. Other treatments, such as irrigation, have also been tested and have had some successes, some mixed results too. There in my hand, I'm actually holding a dry water that's a slow release irrigation gel that has shown effectiveness for some species. Another technique that has been tested is using cuttings, and this has the advantage of not needing to collect seed, but rather employing the clonal reproduction mechanisms of some plant species. And in this case, this was for beaver tail cactus to revegetate a severely disturbed rock mine area that was still pretty barren there, shown on the left. And what was really neat about this study is just taking cactus pads and transplanting them. We achieved really good survival, even though it was extremely drought conditions, and there was 2 times greater survival if plants were placed in nurse rock shelters there, shown in the right-hand photo. And I really like treatments like this because it's kind of creative and employs the use of on-site materials so that we're not having to bring plastics or other kind of artificial materials to the sites. The next restoration practice was seeding, and this was evaluated in 13 studies with 44 species, and the outcomes were highly variable. One of the successful studies was using seeding desert plantain, which is an annual native annual forb, and it's a really important forage species for desert tortoises. And the key to success for this particular species in this study was actually pelletizing and enclosing the seeds in a protective coating. And so that's shown on the bottom left photo there, where there's kind of the whitish coated seeds. And even though this is an annual plant, that treatment resulted in greater plantain densities for several years. And this is one of the studies that we'll be returning to, in this case, about 10 years later, that I'll show in one of the ongoing projects here at the end of the presentation. The next treatment, assisted natural regeneration, and this is trying to help organisms overcome barriers to regeneration. And so often, many years, we see huge numbers of perennial plant seedlings that will appear, but many or all of them will end up dying any given year. And so the intention here is, can we locate some of these seedlings and apply treatments to enable them to then survive? And again, a big advantage of this is that we're automatically using local genetics if we're relying on natural regeneration, and we don't have to go through the huge process of collecting seed, potentially propagating plants for long time periods in the nursery, and then bringing them back out to restoration sites. Instead, we're utilizing the on-site material. And this study had pretty mixed results. There were trade-offs in terms of treatments applied for survival and growth of creosote. And the major conclusion from this was that this method has potential, but we really need more work to figure out which species and which treatments this might work in. And so the next category of treatments was environmental site restoration. And in this case, showing you an example for using vertical mulch. And this is basically planting dead plant material intended to simulate some of the functions of live plants, with some of those functions perhaps being limiting soil erosion, trapping seeds, providing shade, kind of some of that nurse plant type effect. And in this study, the plant there next, or I should say the vertical mulch structure there labeled vertical mulch, that's actually not a live plant. It's these are dead stalks that we collected from live big Aida grass, a native perennial grass, and then put them in the ground trying to simulate a native plant. And again, we could use branches and so forth from woody shrubs as well to do this. And this was done in the context of a bet hedging project that involved vertical mulching and different microtopographic feature creation as well, and then also outplanting too. With the bet hedging being trying to develop and implement multiple treatment types with the intention that if conditions are harsh, that at least one of the treatments will work to avert an overall project failure. And that was pretty fortuitous because it just so happened that the year that we implemented this project was actually the driest in about the last 50 years. And so despite giving irrigation to outplants, all 144 outplants died. But the abiotic treatments, including the vertical mulch, did provide some of the functions successfully that we were hoping to achieve, such as limiting soil erosion. And the graph on the bottom right shows that during a couple years, the vertical mulch treatment ended up accumulating soil. So soil erosion was not occurring. We were actually gaining soil, whereas the controls doing nothing had continued soil erosion. And I also want to mention too that in addition to limiting soil erosion to maintain site productivity on the site, This study was done south of Joshua Tree National Park, and it's a heavily disturbed area. And on really windy days, when we have wind from the south and southwest, and if you see some of the dust storms coming kind of from the south, some of that dust very well could be from this area. So, trying to limit soil erosion was a very important function that was able to be achieved despite the really inhospitable conditions. So the next example treatment here is reapplying BioCrust, and good follow-up here to Nicole's portion. And the two photos on the right-hand side are a site in Lake Mead National Recreation Area, and the 2009 site was 2009 photo was severely disturbed by road construction maintenance to reroute the road, and was before restoration. And then the 2016 photo, again, exact same location. This is an example of a restoration project that really, kind of magically transformed the landscape. You might be able to match up exactly the different topographic features and so forth in the background and foreground. It is from the same exact location. And this involved topsoil salvage and reapplication, salvaging perennial plants and reapplying them, and then also inoculating the site with biocrust. And as shown on the left-hand graph, the biocrust inoculation was really important for enhancing soil stability. So restoration application of some of what Nicole was mentioning. We also examined and summarized different finances and logistics of habitat restoration techniques, and costs varied tremendously from $100,000 per hectare or more to much lower costs of $1,000, $2,000 per hectare. And also want to mention too, these costs are for doing restoration on an entire 1 hectare of land. We might do things like have 0.1 hectare patches, like, and have 10 of them, right? And then perhaps be able to apply restoration across a much broader area, maybe hundreds of hectares, for example, if we're trying to do outplanting in different small patches, as I was mentioning earlier. And I also feel that in terms of restoration costs, this is another area where research can be very helpful, because if we can develop techniques to reduce these costs, it can be tremendous in terms of enabling restoration to proceed cost effectively where it might not otherwise. One example could be if we can even reduce the amount of time that plants need to be propagated in a nursery. Say if we can cut that or shorten that by 1 month, imagine the cost savings in terms of the resources saved. And to that end, just conclude here with a couple slides on some ongoing projects. This was a project supported by Southern Nevada District, Bureau of Land Management Office, and we tested different container types and growing media and how that could affect outplant quality. And so the left photo shows different types of containers. Some of these are even biodegradable, with the advantage then that they could be potentially outplanted in the field with the seedling in them to, again, enhance efficiency, but also limit root disturbance as well. And then the right-hand photo shows a restoration site near Needles, California. And for that, outplants, in this case, it was white bursage, that had been grown in fine textured growing medium, had about twice the plant survival than if plants had been grown in coarse textured medium in the nursery. And so, we could have different results, of course, for different species and different types of sites. But this was one example where the way that plants were propagated in the nursery could then influence their subsequent survival and performance in the field. And just a little excerpt there showing the title there on the top left. We're working on a series of manuscripts from this, and Jonathan P. Smith, or J.J. Smith, with the Southern Nevada District of BLM, is just one of the examples where we really enjoy including land managers as co-authors on publications, and we greatly value that opportunity and greatly value the input and help that they can provide in terms of preparing the best scientific manuscripts that we can in restoration. Another project that we're implementing literally in the next couple of weeks is a retrospective restoration assessment supported by Clark County Desert Conservation Program. And with this, Our goal is to return to restoration sites shown on the map throughout Clark County, some of which are 20 some years old, and see which treatments have worked where. And the really unique feature of this is that we can have a more long-term perspective, because so often our research or agency monitoring spans just a couple years, and we're left with questions such as, what if there was a treatment that didn't seem to work within the first, say, 3 years? What is it now after, say, a 20-year period? Has it worked? Or likewise, what if there's something that seems to have worked initially? Is it still working, once it's had time to go through different drought cycles and so forth? And so this project will provide a really unique opportunity to do that. And with that, I'm ready to turn it over to Dr. Petrie.
Great, thanks everyone. I hope you can all hear me. Say something if you can't. I'm in the office down the hall, so I'm gonna have to present my presentation separately, and I think we should be in good shape here. Let's see, we'll do presentation. All right, I presume everybody can see this. So hi, I am Dr. Matthew Petrie. I am an assistant professor here in the School of Life Sciences at UNLV, and I am a broadly trained natural scientist, ecosystem scientist. I have some background in environmental science and physical geography, as well as biology and ecology. My background in research spans everything from tallgrass prairie in Kansas to desert grasslands and shrublands of our three major U.S. deserts, woodlands, and forests. And kind of along with my broad interest in ecosystems, my research is also very broadly focused. I am very interested in how mechanistic and process-based understanding of how ecosystems function can point us as scientists and land managers towards understanding the most important changes that they may experience, how those changes would come about, and additionally what we can do as humans to reverse or delay meaningful negative change to the ecosystems that we care about in the West. So along those lines, different research techniques give us different pieces of the puzzle that we need to understand ecological change in our region, especially in the future. So my research includes things like field characterizations, ground and satellite remote sensing, manipulative field experiments, meteorological and environmental instrumentation, and also using field collected and experimentally collected data in computational modeling and climate change forecasting. In the next few slides, I'm going to talk a little bit about some of my ongoing research projects here in Southern Nevada that employ some of these techniques. My lab, since I arrived in Southern Nevada in 2018, has deployed 2 instrumented meteorological and environmental eco-hydrological monitoring gradients, one in Southern Nevada in the Spring Mountains, and one in central Arizona at the Sierra Ancha Experimental Forest. Our instruments collect your basic meteorological data such as solar radiation and precipitation, wind speed, relative humidity, things like that, while also collecting some important biophysical variables like soil moisture at multiple depths, soil surface temperatures, photosynthetically active radiation, and things like this. One of the advantages that we have to using these stations is they are a relatively low cost way to obtain high quality data. So for example, using these sort of lower cost instruments, we can deploy data collection continuously at relatively low elevational steps. So our elevational steps are only 200 meters in the Spring Mountains. So we actually have multiple stations within pinyon juniper woodlands all the way up into ponderosa pine and mixed conifer forest. In addition to just doing data collection, which supports a lot of our research goals, we have also focused on characterizing the different ecosystems across these gradients, especially in the Spring and Sheep Mountains of southern Nevada, and implementing the data as parameterizations in computational models. So I do some modeling in-house in my lab and also have a long-term collaboration with the U.S. Geological Survey to develop new research questions that can be implemented in computational models like the soil WAP model, which I'm showing you the diagram of here on the right. It's a process-based, try to do all the physics in an ecosystem type model. And what we can utilize this model for is not only sort of developing better understanding and characterizations of ecosystems in the present, but we've also incorporated a full suite of climate models. So we can do a lot of ecological and climate change forecasting in southern Nevada using these techniques. For just a brief example of how some of this data collection has been useful, so I'm showing you meteorological estimates for a number of characterized sites in the Spring and Sheep Mountains of southern Nevada. And you'll see that I have three ecotypes here in this table. These are ecosystem types. We have mixed conifer forests, ponderosa pine dominated forests, and pinyon juniper woodlands. And what in this table, you'll see we have estimates of cool season precipitation in millimeters, both, and then additionally on the right, warm season precipitation. So I'm showing you results split between the cool season and the warm season for our southern Nevada region. Also, cool season and warm season EP, which is potential evapotranspiration. That is, you can think of it as how much water could be evaporated by the atmospheric environment in these different ecosystem types. That's not exactly what it is, but it's pretty close. And then cool season and warm season standardized precipitation and evapotranspiration index, which is just precipitation minus potential evapotranspiration, gives you an idea of meteorological water deficit. And what I want to highlight in this slide is we are experiencing a really unique meteorological period here in southern Nevada, where, as you see highlighted in green in the warm season and cool season precipitation parts of the table, precipitation in our region is relatively high compared to the past 80 years, and cool season and warm season potential evapotranspiration is also relatively high. That is, temperatures have increased in many cases significantly in the mountains of southern Nevada over the last 80 years, but have largely been offset by increases in precipitation, such that we are actually, from a meteorological standpoint, kind of on average for these ecosystem types, so not everywhere, not all the time, actually somewhat meteorologically less water limited than we were in, for example, the 1981 to 2000 period, highlighting, in my opinion, that we are sitting in a really unique time window for climate adaptation management, at least at higher elevations in southern Nevada, because we are not yet experiencing the potentially severe impacts of temperature increases in our region. Additionally, changing directions a little bit, my lab is highlighting southern Nevada as an important place to understand natural forest regeneration. So a component of my research program is understanding the demographic process of seed production and germination and seedling survival to adulthood in undisturbed ponderosa pine forests. So these are forests that may have experienced management or logging in the past, but have not experienced a recent stand replacing disturbance. My lab has conducted a large number of field characterizations and modeling simulations for managed and unmanaged forests in different locations of the southwestern United States, and unfortunately has found that as many as 30% of managed and unmanaged forests have not experienced any successful natural regeneration in the past 2 decades. So there's potential for there being a new emergent threat to ponderosa pine forests through the regeneration or reproductive niche. Now, in addition to characterizing why these regeneration failures have occurred, you'll see here this sort of heat map figure towards the middle illustrates sort of the conditions that are more severe in sites that have not experienced regeneration. So higher air temperatures, more growing degree days, heat loading in these ecosystems is resulting in, or at least has resulted in 2 decades of regeneration failure, largely at forest locations at lower latitudes, suggesting a potential for latitudinal expansion of regeneration failure in a warming environment. However, additionally, our research has focused on how past human management interventions, broadly classified because we went to so many places and they were doing so many different things as no management, thinning, and burning, how human management has increased regeneration in forests of the southwestern United States. Now, the cool thing that's going on in southern Nevada is this is the place where we're working to try to determine how human planning can reverse these regeneration failures. So the National Science Foundation funded my lab for a 3-year study quantifying the microclimates of planted tree mortality and survival in southern Nevada. So we're currently in the final year of this study where we're using lots of instrumentation, even some ground-based remote sensing, where you can see we're doing some spectral imagery of planted ponderosa pine seedlings, soil moisture, and microclimate environmental instrumentation to determine the conditions that lead to tree survival versus tree mortality in sort of different forest types of the Spring Mountains. What's cool is now that we're entering into year 3, we're starting to get some positive results. So last year was favorable, but we also were able to achieve as high as 70 and 90% survival of planted just literal ponderosa pine seedling plugs last year. So we're starting to figure out what the microclimate characteristics are that allow for survival and how we can adjust planting date to make those things occur. Now, what I haven't talked about here is this graph at the bottom left. An additional study we're conducting in the Spring Mountains and some of our other forested sites is taking the biotic focused approach to better understanding regeneration as a function of hydraulic stress tolerance of seedling and juvenile ponderosa pine conduits. So the P50 value is the soil water potential at which 50% of xylem conduits become cavitated in different, what you're seeing here, ponderosa pine populations of the southwestern United States. In panel A, you're looking at adult P50 values in northern Arizona, NAZ, the Front Range of Colorado and northern Colorado, where adult trees in northern Colorado become embolized at a lower soil water potential, okay, illustrating that they are more sensitive to hydraulic stress or less stress tolerant. And in panel B, you have results from juveniles across the southwestern United States. And what I'd like to highlight here is, once again, southern Nevada is doing something really cool. There is potential that the populations of ponderosa pines that are growing in southern Nevada may be distinct in their stress tolerance from some other populations in the southwestern United States. And moving on, as a final example, trained in desert ecology, we're fortunately also doing some cool desert work. So just in the last year or so, we have instrumented and started collecting data at the Gemini photovoltaic facility, kind of north of Las Vegas. And what we're doing here is taking an ecosystem-focused approach to understanding the different microclimates that exist in these large photovoltaic facilities. And I'm highlighting some differences in air temperature and soil surface temperature and soil temperature below ground between desert environments inside and outside the solar facility. So we're very much still in the data collection phase with this work, but we'll hopefully have some cool research results soon. So I only have a few more minutes here, but what I want to try to do is kind of tie Nicole and Scott and my perspectives on research in Southern Nevada together and talk about why it's important. Maybe mostly from my perspective, but hopefully some things that they would also agree with as well. The first thing is that I'm using just my climate diagrams from forests as an example. Southern Nevada is uniquely situated to inform the potential for warmer and drier summers in the southwestern United States. You know, we have no or very low monsoon precipitation with a relatively strong cool season, relatively high snowfall. And much of the southwestern United States, what are they worried about? They're worried about getting hotter and drier in the future. We can inform through our research and our management activities what other scientists throughout our region should be thinking about. Additionally, the converse of that is a great deal of ecological insight comes from research hotspots that are not fully representative of our region. And our unique soils and ecosystems and climatic conditions mean that, from my perspective, many of the insights that we need in Southern Nevada are things that we need to be developing ourselves. Things like the USDA Experimental Forests and Ranges, not in Southern Nevada. Long-Term Agroecosystems Research Network, not in Southern Nevada. LTER Network, not in Southern Nevada. NEON Network completely ignores the Mojave Desert and ecosystems of Southern Nevada. However, in Southern Nevada, we have the opportunity to get in front of some aspects of ecological change by working together. So my top figure I pulled from a news article, we're experiencing increasing wildfire in the Mojave Desert. It's probably something that we need better restoration information to reverse, to the degree we can, some of these expanding disturbances. More on the bright side, in these middle pictures, the pinyon pine and juniper die-off that is occurring on the left two images in places like New Mexico and Arizona is largely not occurring here in Southern Nevada. We don't yet know why, but if we can understand why, we might be able to keep these declines from occurring. Also, from the wildfire perspective, there have been severe wildfires in Southern Nevada that have impacted our ecosystems, but they have not been as severe and they have not occurred as much recently as they have in other parts of the southwestern United States. Maybe this is something that together we can work to keep these things from occurring and to make our ecosystems more resistant and resilient to the types of change and threats that they could experience in the future. So that's all I have for you guys. And thank you for listening to me ramble for a few minutes. And I think we have a little bit of time for chatting about questions.
Yes, thank you all. It was a terrific trio of talks. We really appreciate your time.